And remember, you have nearly 900 microliter liquid as well there. Let's use the green color. This is the liquid phase. This is supernatant and white pellet. Now this white pellet is, you remember we said we have to get rid of the genomic DNA because we only want purified plasmid. So all the genomic DNA is trapped in the white mass, protein, cell wall, cell membrane. They got settled, they become part of the pellet, and our plasmid, being a small molecule, is still in the liquid phase. Okay. What we do now, we discard, we throw away the pellet and transfer our supernatant to a fresh tube. So we transfer this to fresh tube, the liquid phase, the supernatant, because that is what contains our plasmid. Okay? In the soluble form. Everything else is gone. We got rid of cell wall, cell membrane, genomic DNA, all the proteins, ribosome complexes, whatever. Now, this supernatant, let me draw it. So let's give yellow color to the supernatant because the pellet was white, so I wanted a different color. So this supernatant contains our plasmid. Our plasmid was also, let's say, yellow there. We have gotten rid of everything else, but now we need to concentrate our plasmid because it's in 900 microliter, it will be too diluted. We can't do any analysis there. And there are still many salts. You remember we have SDS there, we have alkali there, we have EDTA there. EDTA is going to inhibit our all future enzymatic restriction, enzyme reaction. So we have to precipitate our plasmid DNA from this. So what we do, I have to draw it again. So this is the supernatant after spinning down the neutralization solution, and after the neutralization step, what we do, we simply add, so in order to precipitate DNA in a solution, we normally routinely use ethanol precipitation method, or we, in case of alkaline lysis, because volumes are huge, we use isopropanol. Okay? If you use ethanol, we normally use 2.5 times the volumes. If it's 900 microliter, the 2.5 times of 900 microliter will be a lot, and that cannot be accommodated in the Eppendorf tube. So isopropanol is normally used 0.6 times volumes, okay, much smaller volume, and it does the same job. So what is the concept of precipitation? What has happened? You remember here, you have acetate ions in the solution. You have the potassium as well. Normally when we use ethanol precipitation, we use sodium acetate in the solution. And what happens, the DNA molecule, the phosphate backbone, it interacts with the sodium ions as well. It makes it heavier. You add isopropanol or ethanol. Okay. These are dehydrating agents. They suck away all the water, and DNA, being heavier molecule, once you will add isopropanol, it is followed by spinning or centrifugation at 14,000 RPM, again for 10 minutes. And what is going to happen, you will see clear supernatant, and you will see a very beautiful tear-shaped, a pellet like this, a tear-shaped pellet at the bottom of your Eppendorf tube. So your plasmid got precipitated. So you keep the pellet and you discard the supernatant. Now this contains still salts in it, and what you do after throwing away supernatant, you now wash. The next step is you throw away supernatant and you wash the pellet, plasmid pellet, with 70% ethanol. So 70% ethanol contains 30% water. Pellet still remains insoluble, it does not get dissolved. And the 30% water present in 70% ethanol is actually washing away all the salts and further purifying it, further making it highly purified DNA. And after washing, you again spin down at 14,000 RPM. I usually do 10 minutes because the pellet, you will be at the risk of losing pellet. So you very carefully discard the supernatant, and you will have purified plasmid DNA like a tear-shaped pellet in your Eppendorf. This plasmid DNA you use to do restriction digestion. You use this plasmid DNA for confirmation of your spike gene cloned inside your vector. Okay? And what you do with this, you set up a restriction digestion reaction. Take some amount of DNA. You need to measure the concentration of your plasmid DNA here. How much what you do normally after washing with 70% ethanol, I usually I air dry this, so that all the remnants of ethanol are also gone, because ethanol will be inhibitory for restriction digestion. So after air drying for 10 to 15 minutes, I dissolve this pellet in let's say 20 to 30 microliter of either Tris HCl buffer pH 8, which keeps DNA protected from degradation, or simply in plain autoclaved water, 20 to 30 microliter sterilized double distilled water in which there are no salts. Okay, and your DNA is good to go for decades, centuries, in -20s, -80s. Even DNA is stable at the room temperature for if you have this in the right buffers. This DNA is used to perform restriction digestion to confirm that original question was, we cloned spike gene in general purpose cloning vector, and we wanted to confirm the cloning of spike gene. So after going through this whole protocol, you will finally digest, perform restriction digestion, followed by agarose gel electrophoresis, and that will give you confirmation. Stay tuned, we'll just confirm this plasmid in the next video.